Hey, Biology 2. I want to do a quick overview of Phylum Chordata, the phylum that we are in for you. And I'm going to screen share my Google site from this section. And I've also posted that Google site for those of you who find this interesting and want to just dig a little deeper or see, what I conventionally teach on this. You're welcome to go there and look around, ask me any questions you want. There's going to be this set of information that maybe will take you 30 minutes to get through, and I will probably post one more set of the traditional in-class information for you, and the rest of the expectation is just getting another submission in on your deep map. And then we'll have a little bit of wrap up with your deep map so that you can kind of give a summary and be sure you have kind of the components in order and give me a summary of how it went. Here we go, just a really quick overview of Phylum Chordata. Let me get the screen share on. Okay, so this is from my Google site, and I just want to go through a little bit of it. So you've watched the short video on Cambrian explosion, and hopefully you caught on that Paikia. This dude here. Wow. So the little picture of me is right in the way of my scroll bar. There we go. Okay, so Phylum Chordata, and starting with a subphylum that we are not in. See, it says Tunicata, Cephalic Chordata, and Vertebrata are the three subphyla. And we are vertebrates, so we are in Vertebrata. But look, there are these other two, and these are really tiny phyla. They don't have very many critters in them, and they're very basic critters. And the main one we'd want to look at is one that looks pretty close to this fossil Paikia. So looking at the fossil impression of Paikia from however many fossils of it they have, they notice that there appears to be some structure up along its dorsal aspect, and there's enough detail that they recognize these different components. The NOT stands for the notochord, and I wanted to talk briefly about a notochord. So a notochord in us ends up just being this vestigial disc space between the bones of the vertebrae, and I mentioned this in my notes last time. But the original version of this, and also found in vertebrates embryologically before the bones form in the embryo, there is a solid rod, and the rod is made out of just kind of like our discs are kind of rubbery material. The rod's kind of rubbery material, but it's also got quite a bit of structure to it. It's stiff enough so that let me stop the screen share for a moment. Stop share. So that when it's compressed, it doesn't squish. Like literally, if I were to really push down, I could get some compression, and that keeps our, that actually allows us some flexibility in our backbone without it collapsing. But if we didn't have the bones, which we have, and we only had the notochord, we would still have little bit compression, but we can't telescope, is what we say. So like this dude doesn't have a notochord, and he would shrink all the way down and stretch all the way up, and our bodies don't do that, which is kind of an advantage to us. And then he's blinking. So we don't squish too much, we don't stretch too much, we're able to hold our structure, and that's good. If you think back to the I'm going to put this back on screen share. If you think back to the video of the mantis shrimp, the reason it was able to punch at the snell was because its muscles were holding on to the exoskeleton. We don't have an exoskeleton, so in this chapter of life within our phylum, we have an internal structure for muscles to attach to, and that also helps us keep shape. So it's good to not telescope, because that means we can, as we start attaching muscle, have the strength to powerfully move and not just kind of mush along. And so in Paikia, we also see these striations, and these are muscles that are referred to as myomeres. And the myomeres are muscles, and the muscles attach from the top structure. So attaching from the notochord down the body in strips. And if you think about last time you ate fish, if you think about how if the fish is cooked, not like cooked too much, and it's not a fish stick, which is just mushed fish, but you know, actual fish where you can flake off those layers of muscle. That's pretty much what this is. Okay, it's layers of muscles. Now these are just 2-inch or so little, they're tiny little animals. 2-inch, what, 5 centimeters? Oh, I'm going the wrong way. So they're small animals. And right now I'm talking about a fossil animal, but oh, let me update and move to one of the phyla that exists today. And so of our phylum options up here, this is Cephalic Chordata, brain cord, cephalus head, chordata. So chordate with from head to tail, so cephalic region. There's some brain up here, not a whole lot, but here is the notochord running along the nerve cord and preventing the animal from telescoping. And you can see the red indicates the myomeres, and the myomeres again are the strips of muscle going from the cord down the body. And what those strips of muscle allow it to do is just swim back and forth sideways. And with more flexibility than I can do at that spine, but it's allowing them to swim this way, this way, this way. So the muscles flinch this way, they flinch this way, they flinch this way. And you know, a fish can go quite rapidly. And these are very fish-like. So these are kind of, you can think of them as kind of ancestral to fish. They don't have jaws. They have kind of this little mustachy area here that they can slurp some food into and ingest from pie hole to poo hole, which I don't have drawn. And they have these, they're called gill slits or pharyngeal arches or pharyngeal slits. It all pretty much means the same thing. These are some supportive tissue, kind of stiff like cartilage, that allow for water to move in and out, water currents to move in and out along some very thin tissue where oxygen is taken up. And so, you know, fish have gills and they're taking their oxygen in kind of external to the body, just going in and out of their mouth region, their oral region, without needing to go internal to the body to get the oxygen. Later on, we get the deeper version of that, which is lungs. But here we have these pharyngeal arches, and they will come up as being interesting in just a moment. Okay. So we're going to move on to what we could look at beyond this animal and moving into vertebrates. So some of the first vertebrates you would think about would be fishes. Well, not all fishes have jaws, but the ones we mostly think of have jaws. So how did we get from a thing without jaws to a thing with jaws? Well, if we look at this amphioxus and its muscle attachment and its pharyngeal arches, fishes also have pharyngeal arches, and those are supporting the gill movement, the ability of water to go in and out, which actually helps them also get some food in their mouth. That's part of the main purpose of it in this animal. This animal is so small it doesn't need as much oxygen circulating its body as a fish does. Fish quite active. It's using these gill arches to also get oxygen close to their red blood cells that are then circulating through the body. But they also have jaws. So I want to take a look at jaws. Oh, and look, there's a little human up here too. So we'll come back to that. But let's think about jaws. So here we have a situation where we can imagine here this jaws being kind of shifted. So we're looking at sections of segments, and this has been a theme since we were talking about earthworms. And we have sections of segments kind of consolidating their efforts to become kind of a different thing. And so if you watch the color codes from looking at Hox genes and looking at species that are alive today and looking at fossil record, here is what seems to be the case, is that some of the gill arches that are towards the front head of an animal such as Pikea started to kind of fortify by the genes that coding for them kind of mutating into a collapsed or truncated or merged set of data that made more reinforced, thicker pieces that eventually seem to be forming the jaws of the animal. So there's quite a bit of fossil record evidence for this. And then again, if you follow the trail of Hox genes in living animals today, it seems to be consistent with this idea. So here we have a situation where now we have jaws. Jaws are important because if you can start chasing down stuff with your faster ability to swim, think you myomeres and muscle attachment onto the vertebrae, now you're able to grab and eat bigger things, and you can get your body bigger. And now we start getting into the wacky world of big predators, and so radiation of species really starts to occur. Another place where we can see segmentation down here is the brain. But first, let me come back up to this real quick. Okay, gill slits. I don't got gill slits, but guess what? If you look at early embryos of vertebrates, gill slits are in the embryos. Okay, so we lose them earlier on in development, but they exist. There are pharyngeal arches that appear in embryonical forms of all the vertebrates. Okay, and so this little dude, not so well drawn, was posted on the board when we were talking about segments. So where are our segments? Well, we've got these vertebra. We have our backbone. Those are segments. The ribs coming off show you segments. Remember, segments got truncated in insects and apparent in all chordates too, where we have a thoracic cavity where your ribs are. Below that, you have your abdominal cavity where your digestive organs are, and a little lower down the pelvis where your reproductive organs are. Okay? And so some of the things that make a chordate is having a notochord, not necessarily as an adult. We've got the vestigial notochord, so check, we got a notochord. Having those gill slits, check, as embryos. Mammals have gill slits. And having a dorsal, along the back, hollow nerve cord. Yes, we've got that. And this is I used to list a fourth one. I'm blanking what it is. It's on a list. We'll get it to you. And then continuing the idea of Hox genes turning on segments. So here we have a brain, starting with the spinal cord up to brainstem area, and where basic functions occur. So this medulla oblongata is part of the brainstem, and that's just like keeping your heartbeat regulated, very basic, keep the body alive stuff. And cerebellum is related to your balance and your understanding of where is my body in the world. Okay, my head is up and my tail is down. I know where I am. I can put my finger on my nose without a whole lot of effort because my cerebellum, which is located back here, is working well for me. Optic lobe, that optic for eyes. Cerebrum, we're getting into motor skills like that. I can, when I choose to put my hand towards the screen, I can do that. And quite frankly, if something were to, like if the ceiling panel dropped right now, I'd have the reflex to duck, just those abilities to use my motor skills to keep myself tuned in with the world, to sense what's happening and react to it. And later on, this also becomes more complex, where I can start planning and plotting and deciding what I'm going to do with myself this summer, and making good decisions like, should I bring my face mask with me in case I need to wear at a show? So higher level thinking, like when I go to the grocery store, I can wear this beautiful face mask that I forgot her name, Mulholland. Oh my God, Maddie, that Maddie Mulholland made for me. Maddie Mulholland, sewing expert. Okay, so these are, anyway, looking back at this brain, you can see the definite segments. And what I want to point out, because I'm going to come way up to the top now, brains. It's cut off, but this one along the top to the right of the mouse is phylum Mollusca. This is a snail. I'm sorry the top's cut off. But here they have their nerves not ventrally, not dorsally, and snails have ganglia throughout their body. And so ganglia, 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 ganglia. Segment, 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 segment. With us, that segmentation collapsed more towards the front of the body, say in the head part of those merged segments, and growing bigger. So it's like the same theme as what I was talking about with the jaws. And those pieces, I don't know if this is color coded the same as what I showed you before, but here's brainstem, here's the cerebellum, this is the cerebral area. So you can't see all the different sections, but if that I showed before, but they're in there. But if you just look, we can do our comparative anatomy with the, is that a duck, goose? It's a goose. The same segments in the lowly little frog. He's not so lowly. Hi, little frog. And they showed it in the mouse as well, but again, that part's cut off. Okay, so we can start looking at those comparisons, and it makes it a lot easier to see how we can be relatives with these rather different and maybe not so different animals in us. I love that. I love being related to. I love feeling. It feels like an extended family. I can use all the family I can get. Like not all the time in my face, but I love to feel that connection. And then I wanted to show you that we can do that with even looking at the skull. And so this is just running the idea of how we go from having all these arches. That fish don't have as many arches. They have fewer arches, and it seems that the first several arches become compounded into or merged into jaw structure. So I'm going to let this run so you can see that. The blue is representing some brain matter. Yeah. But we can watch these jaws forming, and we'll start also seeing the skull develop. I just slowed this way the heck down because it ran too fast. Now I'm thinking it might be a little too slow. Speed it up so I can see my face over here to not quite normal, a little faster though. Okay. So you're going to start seeing bone structure come over it. And so as species start getting skulls to protect their ever-increasing brains, we see in fishes a whole lot of bones, and we see in amphibians and reptiles not as many, but quite a few. But we can follow, like this particular bone has a name. We can follow the merging of the bones as we start looking towards mammals that have fewer bones, but we can always say, well, this is a particular bone, this is a particular bone, and compare across species. And then one more place to look at segmentation and it taking different forms in different species is if we were to look at arms. So I'm going to take you to this. So within vertebrates, once we get to the place that we've moved off of these subphylum Cephalic Chordata that doesn't have bones but has the gill slits, and then we move on to fishes with their jaws and humans that don't need gill slits because they don't need to get their oxygen in that way because it's been internalized into lungs. So instead we start seeing Hox genes with their repetitive behavior forming ribs off of the vertebrae to protect those lungs. We have ever-increasing development of brain. Now, our brain isn't linear, but what happens is this actually starts to, this linear, if this were the spinal cord and brain stem, and these were the top parts, kind of like up to the olfactory bulb, what we see is it's curling over on itself, and our brain starts to take a structure like this, and some of the more ancient parts of the brain actually get wrapped to the inside of that. Such neat stuff. So off of the vertebrae, now we can start getting off the axial skeleton, or the central part of the skeleton. Now we start having limbs forming that are structured and strong enough to draw the animals into land and allow them to motor around. And so let's look at the limbs. We can do some comparative look at parts of some fishes, we call this appendicular skeleton, with the pelvis and shoulder girdles and the bones coming off of those. And most fish, when we're looking at their fins, we can't make this connection, but within the lobe-finned fishes, we can make this connection and start looking for homologous structures. And I love looking at limbs of, we call them tetrapods, 4 limbs, looking at these tetrapod comparisons of the humerus, the upper bone, that's my humerus, and see that we can actually find a homologous structure across all these animals. And it used to be more speculation, and now that we can look at embryology and look at the Hox genes that regulate, it's like, well, of course this is an ulna. I mean, of course it's an ulna, and DNA supports that this is a homologous structure made from similarly coding genes regulated by the same Hox genes that turn on the ulna from species to species. And then just variations of, you know, some people have stumpy fingers and some people are great at playing guitar with their long fingers. And then why wouldn't there be greater diversity among species? So when we look at the bats with the very long fingers, that actually represent a huge proportion of what their wing is. Compared to a bird that doesn't have all those fingies, because some of those structures have collapsed into or merged together to make a stiffened feature to help support the way that they hold their wings. And so it can be such a fun game of looking at mystery solved and mystery yet to be solved. But that's, I think, the big points of looking at some different homologous structures that we know are regulated by Hox genes and being able to start seeing common theme. And so I'm going to leave it there for now. I'll have a Zoom meeting tomorrow. In case you'd like to attend, it'll be our last one. I won't do one during finals week, and might even run it short depending on how many people are there, or it'll be a grand hurrah with a lot of people. But if you have questions for me, I'll be there. If you just want to say hey, I'll be there. And then there won't be a survey because I want to have you put the energy towards wrapping up your deep map and just getting this little bit about vertebrates under your belt before we're finished next week. And so have a good day.